Hello everyone! What we're going to do today is we are going to cover lecture D4, and we're also going to be covering lab 12. They're pretty much covering the same material, so we're going to do both at the same time rather than repeat the material. So let's review what an allele is. Hello! Today we're actually going to be covering two things at the same time. We are going to be covering lecture D4, and we're also going to be covering lab 12. Both of these labs are going to be covering something called Weinberg. We're going to be covering the Weinberg. I might run out of room. E-R-G, the Hardy-Weinberg principle. And we're going to be looking at something called allele frequencies. And the Hardy-Weinberg can actually be a pretty useful principle when we're trying to figure out things like allele frequencies, genotype frequencies, things like that. So let's review what an allele is, and then we'll go from there. So this is the GIMP board that we looked at last time. And remember, this was the complementary base pairs on the DNA. And this was the DNA. And there's different colors here. The different colors are genes. Remember, a gene codes for proteins. So the DNA that codes for proteins is called a gene. And an allele, right here, an allele, is just a variation of the gene. So in this case, you're going to get an allele from your mom and an allele from your dad. In my example, to illustrate this, I am going to use an anolis lizard. So an anolis lizard. Sometimes they sell these in pet stores and they call them chameleons, but they are not. These are anolis lizards. See how cute they are? And in my example, we're going to have a green lizard here and a brown lizard here. In my example here, we're going to say that big B, which is brown, is going to be dominant. And we know there's two ways to get the brown phenotype: homozygous dominant and heterozygous. So we're going to say little b is a recessive allele, green is a recessive phenotype, and we know there's only one genotype to get the green phenotype, and that is homozygous recessive. So I changed my little bees. I didn't like my cursive little bees. So let's say this population actually only has two lizards. One green one and one brown one. So let's say the brown one, put over here, is heterozygous. And there's only one way to have green, right? Homozygous recessive. So in this population, what is the frequency? Let me brown again. What is the frequency of our dominant allele? notice when we have 1, 2, 3, 4. So the frequency of that capital B, dominant allele, is going to give us 1 out of 4, which is going to give us 25%. Does everybody see that? Alright, let's do our recessive allele. The frequency of getting that recessive allele. So we only have 4 possibilities. 1, 2, 3, 4. Out of those 4, 3 of them are little b's. So 3 out of 4. And we're just talking about alleles. And that's going to give us 75%. It's a percent sign. And together, when we add those together, we're going to get 100%. Notice we were just talking about the alleles. Alright, one of the letters. We weren't talking about the genotypes. And we weren't talking about the phenotypes, just the allele frequencies. That's what we're going to be interested in the Hardy-Weinberg. And in the Hardy-Weinberg, they're just going to say that this frequency of the little b, the big B, is P. And we're going to make the recessive allele, the frequency of the recessive allele, as Q. So over here, when we added the frequencies of the dominant allele and the recessive allele, remember, assuming those are the only alleles that are present in the population, we got 100%. So when we add these together, P plus Q, we're going to get 100%. We can say that these equal 1. So we can say that P, frequency of the dominant allele, plus Q, frequency of the recessive allele, equals 100% of the total alleles. And we're going to say that equals 1. Every single time you're going to get a P over a Q. They're the only ones. And now we're going to use that information, that review, to apply the Hardy-Weinberg principle. And again, as I said earlier, the Hardy-Weinberg principle can be useful when we want to figure out allele frequencies, when we want to see frequencies of different genotypes. Yeah, I spelled that right. So Hardy-Weinberg. Now, if you're going to be studying allele frequencies, what we want to do is try to keep the allele frequency stable from generation to generation. So let's look at some things that can keep an allele frequency stable from generation to generation. So in order to apply the Hardy-Weinberg, we have to figure out what are different things that will change them. Alright, so we know one thing could be like selection. So an assumption that we're going to have to make, again, we're going to try to keep our allele frequency stable, is no selection. And we are going to go through each of these. This is just an overall view of it. No selection, no natural selection, no unnatural selection, no selection at all, because if one is reproducing more than the other, like those that have the brown, the dominant allele producing more, right, there can be some selection. So no selection. Another thing, in order to keep our allele frequency stable, would be the mutations. And I'm not going to go through all of them right now. We will go through all of them when we get to our lab book and our lecture material. No mutation. So we're going to have our stable allele frequencies. We can't have one allele changing and turning into another allele. So we're going to keep them stable. Another thing is that it has to be sexually reproducing because we need to have diversity possible. So sexually reproducing. And these things are going to help us keep our allele frequency stable. Sexually reproducing. Reproducing. Again, we're going to go through these. This is for lecture and lab. And it has to be in a large population. So it has to be more than 2. So a large population. So again, we will go through more of these assumptions that we have to make. Large population. So these are some things that are going to help us keep our allele frequencies stable. If you had a small population, it'd be really easy to change the frequencies, like what if they're all brown, or what if they're all green. And let's make another assumption. The assumption is going to be this. There's only going to be these alleles, the dominant allele and the recessive allele, these particular ones that we're studying. So they're going to be 100%, either P or the Q. Remember the P is the dominant allele, the Q is the recessive allele. So we're going to assume that these are the only 2, right? These are the only 2. So you're going to get the P or the Q, or you're going to get the dominant B or the recessive allele, little b. So first let's go to the notes in D4. So detecting microevolution, that is through the Weinberg law. Then we are looking at allele frequencies. And we're talking about the gene pool. A gene pool is just all the genes in a particular population. And we are going to be using this formula. I will talk about the formula a little bit later. First, let's focus on those 5 conditions. And then I'll talk more about the actual Hardy-Weinberg law. So the 5 conditions, besides being large, sexually reproducing, we talked about no mutations, no gene flow, random mating, no genetic drift, and no selection. So let's go now and look at those 5 conditions. Because if those 5 conditions are met, the allele frequencies, therefore the genotype frequencies, will remain constant. Okay, so let's go look at those 5 conditions. So again, the reason why we're looking at these 5 conditions is because we need to keep the allele frequencies stable. So if they're stable, there's going to be no change in the gene pool in a large, sexually reproducing population. So again, we're trying to keep the allele frequencies stable. Mutations. So we can't have any mutations. We can't have one gene becoming another one. Allele frequencies will totally change, right? Now, just because mutation appears may not really affect the population, only if it's heritable, right? So only if it's in the gametes that the offspring receive. Again, not just mutations in general, because these mutations do provide new alleles, but they have to be heritable. mutations of a given gene are rare. Remember all our DNA polymerase going in, fixing those, our DNA police. As a result of a large population, mutations alone does not have much effect in a single generation. And if some new allele rapidly increases its frequency in a population, it's not going to be because the mutation is generating the allele in abundance, but the individuals that have that particular allele are producing more offspring than others. And that could be because of genetic drift, which we will explain, and natural selection. And as we know from meiosis, in sexually reproducing organisms, variation is not primarily due to mutation. It is due to crossing over during meiosis 1, prophase 1, you might call it prometaphase 1. Also, independent assortment, random alignment in metaphase 1, and fertilization. So those are the three primary ways that sexually reproducing organisms are going to have variation. So the mutation itself has little quantitative effect in large populations in a single generation, since mutations at any given locus is actually quite rare. Another assumption that we can make in order to keep our allele frequency stable is no gene flow. This will actually decrease genetic differences between populations by increasing gene pool. So no the I means in, no immigration, no genetic material brought into a new population, no like exit, no emigration. Emigration takes genetic material out of a population into a new population. So let me show you an example. So in this example, we have brown and we have white geese. Population 1 here has brown and white. Population 2 only has brown. Let's say capital A is brown, lowercase a is white. So there's two ways to get brown. It could be homozygous dominant or heterozygous. One way to get white, homozygous recessive. So in population 2, all the brown ones are homozygous. Right? They are this. You get a different color here. All these populations right here, they're all this. Over here, we know the white ones are all two little A's right here. And their brown ones have two, maybe I make it a little bit darker color for that one so you can see it better. So they have two homozygous dominant. They have two heterozygous brown. And they have 3 white ones. Over here we have 3 homozygous dominant. And now what's coming over? Look over here. Sometimes this one's leaving. It's exiting this population and it's going to this one. Notice what it's taking with it. It's 2 alleles. 2 recessive alleles. This one over here has no little a's, no recessive alleles. So it's emigrating, it's leaving this one, entering this one. So that's totally going to be changing the allele frequencies. So gene flow. one organism going to another one, don't just think about that, think about the alleles that it's taking with it. So the gene pool is going to be changing in both of them. So in order to keep the allele frequency stable, we're going to say, no gene flow. No gene flow. That changes allele frequencies. So here's another example of gene flow. So gene flow actually has two principal effects on population. In the short run, gene flow can change allele frequencies in the mixing up population. In the long run, if this takes place over a period of time, both these populations are going to become more similar. So the populations are going to become the same. So it's going to slow down the genetic divergence, black speciation, because the allele frequencies are going to become the same between both populations. The next assumption that we're going to make is no non-random mating, so no non, make it random mating. So individuals have to pair by chance, not according to their genotypes or their phenotypes, so they can't have any type of preference. And we're going to look at some violations of that. Before we do it, let's look at this example right here. Random mating, totally no choice, there's no preference. For example, like sometimes they have like male trees and female trees. So if the male tree, the spores are released, pollen is released. Oh, that was my twin sister. Alright, so let's say we have a male tree and it releases its pollen. It just goes wherever the wind takes it. It doesn't go blowing the wind like, hey, turn around, look at that one, yeah, I like that one. I like that female tree. It was cute. It doesn't do that, just wherever the wind takes it. So, no preference. So violation of random mating would be inbreeding. Allele frequencies aren't going to change. Proportions of the heterozygote are going to decrease, but the proportions of the homozygotes are going to increase. It usually is going to increase the frequency of recessive abnormalities because recessive alleles that are lethal or mutations, if they're by themselves, they're okay, but with inbreeding, there's a much, much greater chance that they're going to meet up with another homozygous allele and produce a homozygote with those two recessive lethal abnormalities, whatever they are. So inbreeding between relatives can give the homozygous recessive allele a greater probability of showing up rather than just by chance. A lot of the purebreds, they can have physical problems because of the homozygous recessive alleles meeting up. So the assumption we're looking at is random mating. In random mating, individuals are going to pair completely due to chance, not according to any type of preference, not according to their genotype or their phenotype. So we saw that with inbreeding, that the alleles are going to get together to a greater extent than by chance. Also, another violation of random mating is something called assortative mating, where individuals mate with those of the same phenotype. Some type of preference. Tall people seem to prefer to mate with tall people, or maybe the opposite. people of the same race, same background. They tend to prefer to mate with those of the same race or the same background. So that violates random mating because there is some type of preference. So they violate it because they do change the gene frequencies, the allele frequencies. So random mating, mate choice is independent of phenotype or genotype. There's two types of assortative mating, positive. The mate choices depend on the similarity of the phenotype. Negative, are they going to choose the opposite? Inbreeding, mating with relatives at a rate greater than expected by chance. So all these violate our assumption that we're trying to get is that all the allele frequencies are the same. We're trying to keep the allele frequencies stable. So in random mating, that will keep them stable. Another assumption to keep our allele frequencies stable from generation to generation is that there cannot be anything called what we call genetic drift. There's two kinds of genetic drift. One is founder effect, and another one is bottleneck. So changes in allele frequencies of a gene pool due to chance. That's the key word, chance. And we're going to talk more about that when we actually get to the lab. Keeping allele frequencies chance due to chance. And this increases genetic differences between populations. It's going to decrease the gene pool. And I will show you an example with those lizards that I showed you at first. And here's another example of a founder effect. Someone founding a colony or something. Rare alleles or combinations of alleles occur at a higher frequency in a population isolated from the general population. And here's an example of the Amish of Lancaster County, Pennsylvania. Today, as many as 1 in 14 individuals carry a recessive allele that causes an unusual form of dwarfism. It affects only the lower arms and lower legs, and also carries alleles for polydactylism, extra fingers. In the population at large, only 1 in 1,000 individual has this particular allele. But all the Amish with this genetic syndrome can actually trace their ancestry to a single couple who immigrated to Pennsylvania in 1744. And the reason why it was so widespread is because they had very large families. And the rare allele became concentrated, increased in that population. Here's an example of genetic drift. Again, it refers to chance. So refers to changes in the allele frequencies of a gene pool due to chance alone. And it is going to be more obvious in small populations. For example, look at these bugs, these frogs. Brown ones, green ones. So in this case, they just died by chance. Maybe they're under a big rock fell and smashed those ones. And so all the ones that were left are green ones. So, in this case, no more brown ones in the whole population. They're wiped out. Let's assume that was the recessive trait. Bugs over here. Green ones here, brown ones here. Just happened to step on these ones, so these ones got stepped on. Totally by chance, the person just stepped on them there. Stepped on that one. Flat. Okay. So it's just by chance. And chance alone, that actually is going to be changing the allele frequencies as you might imagine. So it's going to be changing the alleles, the allele frequencies. So genetic drift has two types, founder effect and bottleneck. So bottleneck is where a population almost goes extinct. And we're talking about when it was chance alone that determined which one survived. Example for today is cheetahs. Now, mass extinction. Today, cheetahs suffer from infertility because of the intense inbreeding that occurred after the bottleneck. So only the few that remained, they had to interbreed. So because of that, only about 0.07% of the population's gene loci are heterozygous. And they need at least 20 generations of inbreeding for that to take place. So a lot of genetic variation is gone. The immune system of one cheetah will not reject a skin graft from a totally unrelated cheetah. And because of inbreeding, the males on average have less than 10% the quantity of sperm of lions and tigers, and 70% of that sperm is defective because of the inbreeding. Because they're all so similar, they may lack the capability to adapt to a changing environment. So if there's a disease that can wipe one, it can wipe all of them. And they have decreased reproductive capacity because of the inbreeding. So they call it a population bottleneck. If you look at this, I think this is a good illustration. Here we have a large population, right here. Here's the bottleneck right there. Almost wiped out. See, almost wiped out there. All these different colors, they're gone. Only these particular colors were left. So a lot of the variation is gone. There's our cheetahs. All of them have this. So the allele frequencies were not kept stable. Our last assumption before we go to the lab book is no natural selection. So that means differential reproduction. And we'll talk about survival of the fittest later when we get to our lab. So no selection. That means all genotypes can survive and reproduce equally well. And let's go look at a very famous example of natural selection. Make sure you know the example. So the famous example of natural selection is the peppered moth in England. In the beginning of the Industrial Revolution in England, coal burning produced soot that covered the countryside in many areas. Look at all that soot coming out. All that pollution. And we're going to look at these. Before you can really understand the example, you need to know what lichen is. That C sounds like a K. You probably have seen lichen even if you don't know what it is. It's a complex life form. It's a symbiosis partnership of two separate organisms, a fungus and an algae. You see probably on bark, some kind of yellowish, whitish. It can even grow on rocks. So lichen is very, very sensitive to pollution. So if there's not very much pollution, you're going to have lichens. If there's a lot of pollution, lichens are going to die. So let's look at the example. Bigger. So in my picture that I drew for you over here, this is pre-Industrial Revolution. So this white here, that's the bark covered with lichens. You can see it down here. That's bark of the trees covered with lichens. Notice the moths are blending in really well with the lichens. You probably can't even see it. And birds actually like to eat these particular moths that we're going to talk about. So moths. Over here, let's say the big B is dark, little b is light. So lichen is covering all the bark of the trunks of the trees. Now birds that come trying to eat the moths, which ones are they going to be able to see better? They're going to be able to see the dark ones better. So they're going to eat the dark ones. And the light ones are going to survive. So look down here. that will increase the recessive allele and decrease the dominant allele. The dark actually will become rare because all the dark ones are going to be eaten. Then, Industrial Revolution comes and it kills all the lichens. See down here. So my purple, that's actually just the natural bark color, which we can now see because all the lichens are gone. So now we can actually see the dark tree bark. Now, if you're a bird here, you can actually see the light ones better. See, look down here in the bottom picture. You can actually see the light ones, and you can hardly see the dark one. There's a dark one right there. You can hardly even see it. So the dark are going to survive. The light ones are going to get eaten. And what's going to happen? Decrease recessive alleles, increase dominant alleles. And even though every single light-colored moth is going to be eaten, you're still going to get light moths if we can cross two heterozygotes, because that little bee is going to get protected by the heterozygote. Alright, so this is our example of a natural selection. So let's review the assumptions of the Hardy-Weinberg equilibrium. Equilibrium meaning we need to keep our allele frequencies stable. And they're actually missing one here. So no selection. No natural selection. All phenotypes can survive and reproduce equally well. No mutations. So no production, no introduction of any type of new alleles due to mutations. No migration, or gene flow is what we call it. No immigration, no emigration. Has to be a large population and sexually reproducing population. Has to have random mating. Mate choice is completely independent of any type of preference, independent of any type of genotype or phenotype. No inbreeding, no assortative mating. And what's the one that's missing? And no genetic drift. No changes in allele frequencies due to chance alone. Alright, so these are the assumptions. And I was going to go right into lab, but actually just have a little bit more on lectures on this next part. Because on speciation is just for lecture, not for lab. And then after that, we will go into material for lab. So everything we've done so far, all these different assumptions, P's and Q's, all that was for lecture and lab. So the next thing we're going to talk about for lecture is speciation. And anything I skip, you can skip as well. Again, this is only for your lecture. So a species, make sure you know the definition. Organisms that can breed with each other and have fertile offspring. And make sure you know our example, horse and a donkey. Horses and donkeys can mate, and if the horse is a female and the donkey is a male, they'll get a mule. The mules are sterile, so mules cannot produce offspring. If the horse is a male and the donkey is a female, you get something called a hinny, which is also sterile. So even though a horse and a donkey can mate, their offspring are always sterile. So they are not considered to be the same species. Horses and donkeys are not the same species. So looking at these, if the male is a donkey, the female is a donkey, they're always going to give donkeys. If the horse is a male, the male is a horse, female is a donkey, they're going to get a hinny. So here's the mule. If the donkey is a male and the horse is a female, they're going to get a mule. If the female is a horse and the male is a horse, then they're going to get a horse. So let's just focus on these two. So the head of the hinny actually is said to resemble that of a horse, the father, more than it does a mule. And it's supposed to have shorter ears. So the hinny has shorter ears, even though they're still longer than those of horses. And they're supposed to be more horse-like manes and tails than mules. So there are some noticeable differences between them. Again, it depends on which parent is which gender. So they are not considered to be the same species because even though donkeys and horses can mate with the mule right here, so whenever we have a donkey and a horse mating, these are the results. And these do not produce offspring. So a donkey with a donkey will give you a donkey, horse with a horse will give you a horse, horse with a donkey will give you a hinny, alright, donkey with the horse with the donkey will give you this. So that means donkeys and horses are not the same species. Both these offspring cannot produce offspring. Hinnies and mules are sterile. So if they're in the same species, they'll produce offspring that are able to produce offspring. Also, same species are always going to be sharing the same gene pool. Gene flow is going to occur between members of the same species in different populations. Gene flow does not occur between populations of different species. Makes you know what a population is? It's just a group of organisms of the same species, same place, at the same time. Alright, now we're going to look at two ways to get speciation. The one we're going to look at first is the most common type. Make sure you know this is the most common. So speciation splits one species into two or more species where the two new species will not reproduce with each other. Alright, the first one is allopatric speciation. So allopatric from the fatherland meaning fatherland, other fatherland. And this is going to deal with geographic isolation. So geographic isolation. So there's going to be something that's going to be preventing them from actually interacting with each other to reproduce. So populations are separated by a geographic barrier that prevents them from reproducing with each other, and that could be because of genetic drift. Then gene flow is going to stop. So no genes leaving one population going into another. And again, this is the most common type. And let me give you an example. This is an example of allopatric speciation where there is some kind of geographic barrier. Let's look at the first picture, the one on top. Right up here. Notice all these flowers, all these plants here, they all look the same, don't they? So they can pass their DNA, their alleles back and forth. But let's say there's an earthquake or something that allows water, which is a different color here, some blue maybe. So water now, let's see the background water now is going to be able to go this way. And these two sides are separated geographically. They are no longer going to be able to exchange their alleles. And over time, given enough time, notice how different they're going to become. They become two different species. And then let's look at our bottom picture. So now the water has receded, so the water is now left. And they still could overlap right here, but they will no longer reproduce with each other because they have become different species. So again, this is the most common type where there's some kind of geographic barrier. Our other kind of speciation that we're going to look at is called sympatric. Sympatric, that means the same fatherland. So speciation that's taking place in the same area. Members of a single population are going to develop some type of genetic difference that's going to prevent them from reproducing with the parent type. And a very good example of that is polyploidy in plants. Multiplication of chromosome number in certain plants of a single population. Let's take a look at that. So polyploidy. Remember, diploid, 2 sets. Poly means more than 2. So let's say here is a cell. And let's have some spindles. We're saying that this is going to be metaphase of meiosis 1. I'll just make them all, these chromosomes, these guys, all the same color. So it's supposed to be diploid, right? Bivalence in the middle. Now something can cause all these to reproduce, and then instead of having just 2 sets, let's say we have 4. It's supposed to have 2, but we get 4. I'll talk more about that in lecture. But I'm just showing you this illustration that this is actually one way that we can get sympatria. And so these ones with 4 will not be able to reproduce the ones that just have 2. So sympatric speciation, often due to polyploidy in plants. So here we have all one species of plants. And then right here, let's say polyploidy takes place in those that have 4 sets rather than 2 sets, the light green ones, are no longer going to be able to reproduce with color here. These ones, so the normal green ones are no longer going to be able to reproduce with the lighter green ones. And it doesn't take a large area to do this. It doesn't, let's read the whole thing. It does not require a large area to reduce gene flow between parts of a population. Right there, it was instantly changed. So let's go ahead and compare the 2 types. Allopatric, this is the most common type. Notice there's one kind of geographic isolating factor, a barrier, and they produce 2 different kinds. Allopatric, they're in 2 different locations. Over here, sympatric, they're in the same location, and that could often be through polyploidy. Something happens, and instead of 2 sets of chromosomes, you get 4 sets. And now 2 different kinds in the same location, not able to breed with each other. The last of the material that is just for lecture is adaptive radiation. Make sure you know what this is. The rapid development of many new species from a single ancestral type, which has spread out and become adapted to living in different habitats. And a famous example are the 3 species of finches on the Galapagos Islands. They are believed to have descended from a single finch type. As the parent population increased in number, daughter populations arose on the various islands. Daughter populations were perhaps subjected to founder effect and natural selection. Each population became adapted to the habitat of the island it was on. Over time, populations no longer recognize each other as being members of their own species and do not interbreed. So let's say we have an island here with some finches. They fill up all the territories, and they say, okay, there's no room for you little baby finches. I'm sorry, but you gotta move on. You gotta leave. So little finches leave, and they go and find another island. Let's say that island is a little different than this one, so these ones have to adapt. Let's say a different kind of plants, maybe. Maybe this is more small seeds and this is bigger seeds, so the ones that are adapted or have the DNA for the bigger seeds are going to survive. They fill up all their territories because these finches are actually quite territorial. The territories are quite large. And they say, okay, you filled up all the territories now, you're going to have to go and make your own way. Go to this one. They adapt to this one. Maybe the coloration of the plants are slightly different, so the ones that blend in with the foliation are going to survive better and pass on their DNA. And then go on 13 different islands, so when you get to the very end, they probably won't even recognize these ones over here. So that is called adaptive radiation. Maybe they had to adapt to different types of foods. This one ate leaves. And maybe the original one is this one. Some went to an island that had more buds and fruit. Those that have the DNA for that one, to adapt to that one, they pass on their DNA. Over here, various types. These ones eat insects, so their beaks are a little different. So that could be an example of allopathic speciation. So let's look at these. Allopathic speciation involves physical separation of populations. Sympatric involves reproductive or behavioral separation, so they're in the same location. Allopathic, populations occupy different geographic areas. Sympatric, same geographic areas. Allopathic, example, adaptive radiation of the Galapagos finches. Sympatric, polyploidy in wheat strains. And since it's taking so long, I think I will do the rest of the lab video on a separate video. So all the speciation is only for lecture, but everything else is for lecture and lab.